Pleasure to introduce Dr. Ethan Gyllenhaal. In 2017, they got their Bachelor's of Science from University of Rochester in Ecology and Evolutionary Biology and a minor in Computer Science. And in 2024, they graduated from the University of New Mexico with their PhD in Biology, where they were advised by Dr. Michael Anderson and supported on the NSF GRFP. And now they are an NSF postdoctoral fellow at Texas Tech University, Guns Up Red Raiders, where they were co-advised by Dr. Joe Manthe and Philip Messer. And so I will let Ethan take it away and tell you a little bit about what they've been working on. Let's give them a warm welcome.
Thank you very much. I'm excited to talk to you about some of my work today. It's been such an awesome day getting to see all the collections and meet with all the grad students and other folks in the museum. So I work on island birds, specifically Pacific island birds. I research them to understand the incredible diversity that they show. And the way that they manifest this diversity is something that's pretty interesting. It's through the form of geographic variation, where when you look across an archipelago, you can see variation in anything from the frequency of a melanic color morph in these monarchs to more complex phenotypes with very strong population structure across an archipelago in these dwarf kingfishers, or even complex dynamics of hybridization and secondary sympatry, like in these monarchs. And geographic variation is really important to understand in any system because it's ultimately the raw material for speciation most of the time, where you'll have a population that has isolation occur within it or dispersal to a new area, and over a period of time, phenotypic divergence will form. And eventually, potentially, if secondary contact occurs, you'll have some sort of coexistence or a hybrid zone or even a lineage fusion among those populations. And when I showed you this big picture of island birds, a lot of what you might have thought about for evolution in Pacific Islands are these ones that are known for their coexistence. Ones like Hawaiian honey creepers and Galapagos finches that have very rapidly evolved the ability to coexist ecologically and in terms of reproductive isolation. But ultimately, those are the outliers of a lot of this South Pacific avifauna. Most of them are on less isolated archipelagos, still to the east of New Guinea and Australia, but tend to represent a more homogeneous, co-distributed assemblage that gradually drops off as you get further out into the Pacific. And they have patterns that look less like rapid co-occurrence, more like this, where you'll look in one archipelago, here are the Solomon Islands, and you'll see this incredible plumage diversity across the archipelago, and you'll look 1,000 miles away in Fiji, see a very similar pattern where you have a lot of plumage divergence with no actual secondary sympatry in the entire lineage and the entire clade with intervening archipelagos in there as well. Rather, you see this strict allopatry, right, without co-occurrence. And understanding both why these groups are so widespread and diverse, but also why they don't occur most of the time is one of the focuses of my research. And one thing to understand about this island system is that the way that divergence occurs is likely not a homogeneous process, right? When we have two birds in different islands that look different, it's easy to view them as having always been isolated, right? But ultimately, in oceanic islands that were never connected to continents, that certainly wasn't the case, right? You had birds occupying one island that then colonized another island, not just once, but enough to form a population that doesn't immediately fizzle out due to inbreeding. That population can diverge over time, but unless you have the reduction in dispersal ability, you likely have gene flow not only into that second island, but also backwards towards the first. And this relates to a hypothesis that I'm going to touch on several times here, part of the taxon cycle hypothesis that is linked in with the idea of great speeders, where you have very high dispersal early on in the lifespan of a lineage spanning all these Pacific islands, with very high dispersal and occasional crossing between archipelagos, allowing this colonization. But over time, you have the evolution of reduced dispersal ability for a variety of reasons, resulting in minimal dispersal between archipelagos and little to none within the archipelagos, allowing them to better produce this phenotypic divergence, but likely not entirely being isolated over time. And I'm going to explore all these aspects a little bit in my talk. I also want to talk briefly about the way that I approach understanding geographic variation. Like most people here who work on phylogeography or similar scales, I start with museum specimens, either directly by observing the series or through the work that other researchers have done on them over time. I then tend to apply a standard genomics toolkit that many of you are familiar with, anything from population structure analyses to test for gene flow. But I also incorporate population genetic simulations using programs like SLIM and MS Prime, so I can get a more strong understanding and test the alternate scenarios that might have led to the patterns of geographic variation and population structure that I see on the landscape. And because I've already heard a lot of interest in simulations, both from colleagues and from people here, I want to briefly talk about the kinds that I use and how I use them. These examples are not actually going to be ones that I talk about today. So they're little teasers for other work of mine. I use coalescence simulations sometimes, where I can ask stuff like how gene flow can impact phylogenetic inference, where our population genetic summary statistics change over longer periods of time. These are very fast, but ultimately you can't incorporate very complex processes. When I need to start getting more complex and adding selection, I'll use non-spatial simulations in SLIM. These can help me ask questions like how sex-biased gene flow might impact chromosomal divergence, like the plot you see there. Or it could help me address how genetic variation might be lost in the span of hundreds of years for given population decline, or even not be lost in that given span of time. I also use spatial SLIM for ultimately the vast majority of my work, and that's what you're going to hear today when I talk about simulations. It's where we track individuals not just through generations in a panmictic landscape, but across a discrete landscape, oftentimes through a geographic map. I could ask questions like what colonization route is most likely, given the geography and some assumptions about dispersal. But I want to emphasize quickly that these simulations do not provide you a singular answer on their own. They can provide additional support or clarify if alternate hypotheses can even be disentangled with the data that you have at hand. And they can be used more directly for empirical inference through machine learning classifiers as input for them. Although I haven't worked on that personally, but it's something that a lot of people are very interested in. And it's an emerging use for them that I'm looking forward to seeing how it develops. I also want to quickly mention how I work on modeling island gene flow in these simulations. In the past, I put together a framework where you can get a rough estimate of number of migrants, given a bunch of parameters of island geography and also some parameters for dispersal using an equation that we're not going to go into. But we're going to go into the couple major takeaways that are going to at least briefly come up during this talk. That further islands receive fewer migrants than closer islands, right? That's pretty intuitive. That you get more gene flow from large islands to small islands because you have a larger number of potential migrants in those large islands. You have higher gene flow between islands that run parallel to each other. To show you how two of these work, we're going to look from Guadalcanal towards the island of Makira and Malaita in the Solomon Islands, where Makira is a little bit further than Malaita, but more importantly, it occupies much less of the horizon, meaning that it makes a much worse target for birds dispersing out from Guadalcanal. And we expect 7 times higher gene flow for a given dispersal distance that I use to produce this number. And you can also see this in terms of the taxa that are shared between them, right? They share a disproportionate number and oftentimes have more closely related populations than you might expect if you were just basing it on island distance and island size alone. I'm not going to talk about the mathematical of these other than the general predictions. I'm going to focus on how I implement this in SLIM, where I have a given probability of what is normal short distance dispersal, just moving within an island, not crossing large water gaps, which happens 99.9% of the time. That 0.01 is this parameter called alpha. I'm going to refer to it as dispersal probability later on. And you get some amount of dispersal at this low frequency where an individual will decide it wants to disperse long distances or a longer distance, choose a random direction and a maximum distance based on our distance decay equation, and then cross that water looking at what is beneath it. And once it encounters land again, it'll land and live in that new island. Or it'll just cross within land, or it'll go into water and die. This means that a lot of these parameters in this equation arise naturally from the map. So that's just kind of covering a couple things you'll see later, right? Now, for what I'm actually going to talk to you about today more explicitly, we're going to look at first what the population genetic dynamics are in archipelago radiations in the Solomons. First, trying to understand what predicts genetic divergence across different taxa in a comparative phylogeographic study, and using a mix of that study and simulations to talk about how genetic diversity varies across archipelagos with respect to island size. Next, I'm going to turn to Fiji, where I aim to understand how phenotypic diversity gets generated in these archipelago radiations. First, looking at how, in general, plumage diversity has evolved in a particular system there, and then understanding what the impact of that divergence is on secondary contact and hybridization in that system. A lot of collaborators to thank for the Solomon Island stuff, but the main ones are folks based out of universities of Kansas, Michigan, Texas Tech, and University of New Mexico. And I showed them on a prior slide, but the Solomons are an archipelago located to the east of New Guinea, relatively high diversity compared to other ones like Fiji that you'll hear about later. And a lot of the reason why they make a great system to study is that you have a very complex geography. You can see here these different colors correspond to the 4 different island groups, with 2 of them, New Georgia and Bukita, having complexities within there. And the Bukita group in particular being very connected during glacial cycles, which is that dark gray that you can see on the map. And you tend to see this pattern in these kind of great speciators that I've talked about, these birds that are very good at generating phenotypic divergence, but not secondary sympatry across these archipelago systems, where you look across, say, the monarchs and see a different phenotype in each of these island groups with some additional variation in the New Georgia group in particular. Same for the dwarf kingfishers, and again for the whistlers, like you saw earlier. One thing that this comes with, particularly for these more deeply diverged taxa, either great speciators or more phenotypically homogeneous ones that still have high genetic divergence, is strong genetic structure among these 4 island groups, with the next strongest break being between parts of the New Georgia group that were separated by a long-term open water channel. And then when you look at the Bukita group, you can see some structure once you zoom into it. Oftentimes more or less reflecting isolation by distance of the current islands. So I wanted to understand what predicts the levels of population genetic divergence in this archipelago using about 16 taxa, both these classic great speciators that have a lot of phenotypic divergence across the archipelago, and ones that are much more homogeneous, not even having subspecies described across this area. I'm going to show you one aspect of this study, which is going to be the relationship between genetic divergence as estimated by mean inter-island group FST, and 2 metrics for dispersal ability. I looked at several other things, but these are what, spoiler, wound up being most important. I used the number of islands that a given lineage is on in the entire northern Melanesian multi-archipelago system from this Fire and Diamond book based on survey data, and hand wing index metric that bird people in the room might be familiar with, that is more or less measuring how long a bird's wing is, which should correlate with its intrinsic dispersal ability. And what we see is going to have first dispersal ability in these 2 categories on the x-axis, where you have more dispersal going from left to right, and then FST on the y-axis, where there is more divergence as you go up explicitly, and based on a set of assumptions, more gene flows you go down that axis. And what we see is a very similar, very strong relationship between dispersal ability in both of these categories and this metric of genetic divergence, which tracks the idea that you have gene flow driving this inter-island group population structure in some capacity. And I want to note, of course, as you might expect, these 2 metrics are very strongly correlated. So disentangling them is a bit tricky. What I think is ultimately going on here is that you have an ability to cross water gaps that is related to not just your intrinsic wing length, wing index, but also a willingness to cross those water gaps that's much harder to quantify. And that cumulative dispersal frequency correlates both with the number of islands that these birds are on, right? Because this difference that you see between taxa is largely driven by occupying very small islands that cannot maintain an independent population for long periods of time. And it then in turn correlates with gene flow between these island groups as well. So it's a bet that every way that you test for the strength of these predictors, the number of islands occupied comes out as a better predictor of genetic divergence. But I think the relationship is a bit more complex, right? Where it's ultimately driven by something we can see on specimens and something that we can't see without extremely good ecological data. The other nice thing about it being a comparative phylogeographic study is it can kind of give us a window into the evolution of this archipelago's avifauna over time. Consistent with that kind of taxon cycle idea that I talked about earlier, we have one example of what seems to be a very recent colonist that is widespread across the archipelago, but has notably reduced net genetic diversity, kind of clear signs of bottlenecks within islands, but very little structure in the phylogeny. What can then happen if this fantail persists over long periods of time, which it seems like it will? It's a very good human commensal. It could get kind of an equilibration of effective population size and clustering, not by any colonization order, but rather by the relative levels of gene flow among these island groups, which as it decreases over time, and you get this stronger divergence, you can get this strong clustering, well-resolved topologies, et cetera, regardless of whether or not it reflects the actual colonization order. One thing that I'm going to mention here, the birds that were not included in this study that occupy the archipelago are ones that have a pattern referred to as relictualization, where they're no longer found across the archipelago. Generally, they're long branches, although not necessarily always. One example is this white-eyed starling, which is specialized on relatively high-quality forests, where the hypothesis is that it's being outcompeted by the metallic starling in the middle of those population structure plots in that area, so it's much more restricted and much more rare. This also encompasses birds like this Melita fantail that are restricted only to the highlands on these islands. We're going to jump to a very different aspect of population genetic theory here, where we're going to go away from population structure and more look at genetic diversity. And this specifically is going to be about the relationship between island size and genetic diversity, which you can probably tell I'm going to relate back to classic island biogeographic theory, namely the relationship between taxonomic diversity and island size, where on smaller islands you have more stochasticity, meaning that you more quickly lose species. And as you might have guessed, you also have more stochasticity leading to faster genetic drift, where you can see as we have larger population sizes, it takes us much longer to lose alleles or have them rise to fixation despite the same starting frequencies. This relationship has been shown in many taxa before, and it suggests some potentially strong predictive power. But we want to explore it across this archipelago where we know that everything is willing and able to cross water gaps because they had to at some point to get there, right? And we're going to look at it in three ways. The first, with this most iconic great speciator, the white eyes that have the most species level taxa in the LSUMNS. You can check out a recently released paper if you want to read more about those. I'm going to briefly cover the summary of the results, so it's pretty straightforward. Then I wanted to look across that same comparative data set to understand how widespread this relationship is. And then a bit of a spoiler, I want to understand when we don't recover this relationship using SLIM in a package developed by former lab mate David Tan called Pleistodist. So for our case study, we had sampling across the Solomon Islands of these white eyes. The New Georgia group with the orange dots in the southwest of the archipelago. There's a ton of described species, and these things are even on remain distinct per prior genetic studies, even on islands that were connected during glacial cycles. So there's reason to believe that they should show this relationship strongly. And I'm going to show you a series of plots where we have an estimate of island size on the x-axis and a diversity metric on the y-axis. When we look at SMC class method estimated historic population size, we see both recent and old long-term population size is strongly correlated with island size. Same with genetic diversity as measured by heterozygosity. We also see evidence for relaxed purifying selection in the archipelago with a very strong negative correlation with the number of transposable elements that taxa seem to have accrued. And with the runs of homozygosity, this conservation genetic metric, although I'll note, even the ones with the most runs of homozygosity do not have enough where it is a clear sign of conservation genetic concern. They're doing fine with respect to inbreeding. And when you look at this and look at only the taxa in the New Georgia group, you can see there a lot of what is driving these strong relationships, right? And I want to quickly kind of make it a bit more complex how we think about gene flow in archipelagos, where when we have this initial colonization followed by high gene flow and then some form of dispersal evolution or lack thereof, right? If we have stasis or if we have complete loss of dispersal, what's likely happening a lot of the time is that you have a decline in dispersal over these periods of time, right? Where you don't have necessarily a total drop off and totally sedentary behavior, but rather a reduction as time progresses, resulting in, say, strong structure between archipelago groups, but minimal within structure, or at least very homogeneous genetic diversity, as we're going to look at here. Where with stasis, we expect no correlation between the two metrics with island size. Where we have very strong lack of dispersal or biological species, we see a very strong relationship like we saw in those white eyes. But for this reduced dispersal situation, we would expect to see kind of a correlation with island group size, but not island size per se, where we have a relatively flat line within the New Georgia group. And that is what we're going to focus on when we're looking across taxa, remembering that the New Georgia group is a lot of what can give us power to detect this relationship in white eyes. And we do indeed detect it. It's even stronger in this comparative data set I'll mention because we calculated island size a bit better using that package placidist I mentioned earlier, taking into account historical island size, not just modern island size. But when we look across the majority of taxa here, there is no significant relationship. There are a couple that do show significant correlation, although I'll note the slopes are much less than in the white eyes. There's a few more with a suggestive relationship and even one with a negative correlation, but that's a very recent colonist of the archipelago, that Willie Wagtail. And when we zoom in on a lot of, especially the great speciators that don't show a strong relationship, we can look at our white eyes and look at them and see that that lack of variation in the New Georgia group is likely what's driving it. This is consistent with the white eyes having this very strong drop-off in gene flow and the partial drop-off in other taxa, like these dwarf kingfishers. However, we should expect that this relationship is a very strong one and not necessarily one that's easy to break. And I wanted to understand that using simulations, where I simulated first island colonization and then divergence using SLIM. I had what I call a synthetic glacial cycle. Rather than lasting 100,000 years, it lasted 20,000 years to make it a bit tougher to recover this relationship in the first place. I also had a lot of total individuals and a varying number of starting individuals that I didn't find to be notably change the results. Where I parameterized that starting population with an empirical allele frequency spectrum from actual white eyes that occupy the New Georgia group. I then had a period of natural dispersal where birds were freely able to go between all the islands in the archipelago randomly. And then I either maintained that over the full course of the simulation, or I had a variable decline in dispersal. And that initial dispersal frequency and that decline are the two things that I varied that you're going to see in these plots. The output for all of this, I then periodically have all the island genetic diversity estimates in a big data frame that I read into R to calculate linear models for all of these, which are going to be in the plots that you see now. They're a bit complicated. So I'm going to use what's going to be a pretty straightforward one to interpret first, where on the x-axis, we see that drop-off multiplier for a given plot. So as we move from the left to right side here, we have no gene flow at the end of the simulation to the same gene flow as the start. 1 means it remains the same, 0 means gene flow fully drops off. Between plots, we're going to see those different starting dispersal distances. And the y-axis here is the first metric, which is just the size diversity relationship r squared, showing that we have strong predictive power most of the time. And I'll note that I think it's 96% of all simulations, regardless of dispersal value, showed a statistically significant correlation between island size and genetic diversity. R squared didn't really vary much across these simulations. It remained high throughout, reflecting the strength of this. And we did indeed see pretty high values, even for some of the not statistically significant ones in the island, in the other taxa, but this likely reflects the relative noise of empirical data compared to our beautiful simulations where we can sample as many individuals as we want. What wound up being a much more informative metric that I think makes sense for why it's better for assessing the homogeneity and genetic diversity is the slope, which we see as we have an increased drop-off multiplier. We have a decreased slope, right? That slope, even though the predictive power remains the same, the slope becomes much weaker as we have more present gene flow. And I want to note that it's only present gene flow. The relationship remains strong if there is high past gene flow and minimal present gene flow, like in here, or even in our highest dispersal levels. It's that ongoing gene flow towards the end of the simulation that really predicts this drop off. And we compare it to our empirical slopes. We see that our white eye has this very high value, these very strong slopes, pretty much matching the simulation, which makes sense because ultimately this simulation is designed around the white eye as success, but everything else is much lower. And taken together with the prior bit, it kind of tells us how gene flow is likely impacting these archipelago taxa that are currently very isolated, and we can easily view them as independent, but there's a lot more homogeneity and connectivity than we might think, kind of suggesting that they're acting more like a metapopulation, where we can see a very strong relationship between the number of islands occupied and FST, showing us how dispersal between island groups can impact genetic homogenization. And also we see this pattern where most taxa seem to be kind of having their genetic diversity homogenized across particularly the Georgia group due to ongoing dispersal across smaller water gaps. We're going to change gears now, switching over to the work that is the center of my postdoc, although a lot of that stuff that I just mentioned, I've only started doing in the past year. This is what the PRFB is actually about. It's about whistlers in Fiji. So there's a lot of collaborators from the Ministry of Forestry in Fiji, Texas Tech, Cornell Computational Biology, and ongoing collaborations with the folks at KU. The Fiji whistlers act as kind of an old-fashioned model for allopatric speciation. If you read any of Ernst Mayr's books, you might have seen this map pop up once or twice, and the mention of Pachycephala several times, where he saw this very unique pattern where Fiji had a very high amount of diversity in whistler phenotypes, ranging from these unbanded birds to these white-throated banded birds, and this seemingly intermediate banded yellow-throated phenotype, as well as a few hybrids. His conclusion was that this was a case of secondary contact between very old lineages. To kind of cut to the chase a little bit, that was likely inaccurate, but we're going to focus on what we can learn. First, about how this archipelago generated extensive plumage variation in a short amount of time, and then understanding what shapes that post-divergence gene flow that we observe. I'm going to first talk about just a summary of the phylogeographic results of this story, which are published in SysBio Spotlights if you're interested, where we have sampling from most of the archipelago. All the important phenotypes are represented, although we unfortunately don't have a couple islands yet, although we're going to be filling that in with whole genome resequencing data shortly. I should also mention later on, although the story that you're going to see in a bit is from RADseq, we do have whole genome resequencing that you'll see later on used in a GWAS. So you don't have to interpret this tree. I'm just going to walk you through the basic story, where we have this kind of laddered stepping stone pattern for these white-throated taxa going from south to north in the archipelago, which seem to have lost that white-throated phenotype at some point in the colonization history before evolving an unbanded phenotype, where there's potentially some period of stasis and either no whistler or a white-throated taxon on Viti Levu, followed by likely facilitated by glacial cycles, secondary contact on the Natewa Peninsula of Vanuatu in the north that I'm going to talk to you about a lot more in a bit. and an invasion of Viti-levu by this bandless taxon, potentially resulting in genetic and ecological swamping of whatever was there. Although we do see signs of admixture with a white-throated taxon on Viti-levu. And in turn, we find evidence of extensive admixture on this island of Ovalau between whatever's on Viti-levu and I'll show you in a bit where it came from, but an unsampled taxon. Starting with that hybrid population on Ovalau, when we look at it instead of in a classic phylogeny with tree mix, which allows us to add gene flow edges, we can see it start out as kind of sister to all of the yellow-throated taxa here. But when we permit gene flow, it winds up having the inference of about 35% of its ancestry from an unsampled thing. That's what kind of that basal thing looks like. Not a good word for it, but whatever. And the rest of it coming from the birds sampled on the immediately adjacent southern part of Viti-levu. And trying to figure, it's interesting to think about what the phenotype of this bird, of this population that was swapped out might have been. And we can try to take some hints from another plumage feature. If I didn't mention, the main way that they vary across the archipelago is in breastband and throat phenotypes. But they also vary in the color of their underparts. You can barely make it out here, but the Ovalau birds and the white-throated birds both have kind of a lemon yellow underpart, whereas the birds in the north part of the archipelago are more orangey. I'm going to focus on the missing pieces of the story in Ovalau and some of the unsampled ones that we're hopefully going to add and resolve what's going on there shortly. So this is Ovalau, consistent with it having a lemon color. You can see on the y-axis, it has a more amount of green reflectance when we have a spectrophotogrammetric measurement of its belly feathers. Whereas one of the parental that is more kind of orangey colored on Coro has a much lower value of that, right? That pretty much is just saying it's lemon yellow, not orangey yellow. What we can see looking at specimens and photos, but getting a bit more quantitative. And when we look at two white-throated taxa that we didn't sample, they show the same pattern, right, where they have this higher level of greenishness to their yellow. But what's really interesting and kind of prevents us from drawing too many conclusions from this, the birds from Viti-levu, that one that we inferred to be admixed with another white-throated population, also have this lemon yellow. So can't truly confirm if it's the white-throated thing that was swamped that has that lemon yellow genotype represented in Ovalau, or if it's just kind of dragged along from the rest of the birds on Viti-levu. So not the most satisfying end of the story, but we're going to focus on my current study system, the hybrid zone in the north part of the archipelago. So one thing about islands is that hybrid zones there are very rare, right? Most of the time, especially in birds, and specifically I'm talking about these more isolated or moderately isolated South Pacific islands. Most of the time, due to the relatively small size of these islands and high dispersibility of birds, you don't get enough consistent parental input to have a traditional hybrid zone. You normally get hybrid populations like I talked about before in Ovalau. That's not the case for these whistlers across Vanuatu. When we look at their phenotypes, we can see that there's tons of intermediates spanning the Natewa Peninsula in the southeast and some outlying islands. And we see the genetic background largely matching this. However, you might notice that we have a big break in the phenotypes, in our sampling, and in the genotypes at the bend of that Natewa Peninsula, which is something that I need to fill in to fully understand what's going on here. And that's what I've been working on this past summer in a scouting and networking trip, and hopefully this upcoming summer for a actual specimen collecting trip, where I went to the communities along this peninsula and looked at the birds there and met with them, particularly with the amazing help of Manasa Vula, the provincial conservation officer who helped me with all of this stuff. And pretty much as I predicted, the birds in this narrow bend in the peninsula are those phenotypes that we missed before, and hopefully the genotypes, although obviously we can't tell yet. Pretty much also confirming something that we didn't know, that a second trait, the amount of yellow in the lores, which you can kind of see in the bottom left photo, that base of the bill, is very yellow in there. Normally, we only saw that on the most unbanded birds, whereas in the hybrid zone core that I was able to go to, we saw it in more intermediate birds or even birds with pretty extensive bands, which was exciting to confirm that they're unlinked, have another thing to look at for the GWASs and whatnot. But what I'm really most interested in is not necessarily the genetic basis, although I am very interested in that. We'll hear about it later. I care a bit more about the geography of the situation, right? And why we have an island hybrid zone here on Vanuatu, because it's normally such a rare phenomenon. And when you look at these islands, you expect the opposite pattern, where we would have more gene flow from the large island to the small island and vice versa. But ultimately, what's unique about Vanuatu is this peninsula in the southeast corner with open water bay that limits gene flow from the quote mainland of the island to that isolated peninsula. And that, I believe, shifts the effective gene flow there. And if there was no channel, we would hypothesize that that gene flow would swamp out new migrants so that you only have intermediates in that far corner of the island. And although I can't fill in the Natewa Bay myself, I can test the hypothesis that it's essential for this contact zone using those simulations in SLIM. This is kind of a dummy simulation. It's not actually one that you're going to see results from because there's a selective benefit to the banded allele here, but just showing you these individuals dispersing across the landscape, having a brief glacial cycle for part of it that we is kind of replicating the more complex history of the region, but we don't need to get too much into that. I'm just going to talk to you about how I ran these simulations. So I pretty much had that going for 100,000 years, and at the end, it's periodically throughout, I would output these pseudo transects going from the west part of the archipelago or of the island and east through that bending peninsula, where I expect that when we look at the frequency of the simulated banded allele, it will increase as you go towards the east with a major inflection point at that bend in the peninsula. How we actually ran these simulations were either with or without this kind of fake glacial cycle, but that's not the emphasis here. The main emphasis is just whether or not we have this filled in channel. We let them run a long time, 100,000 years, for a realistically dense population there. And our big finding is that even without any assortative mating or selective differences to that banded allele, we can produce a pattern with this strong inflection point at that base in the peninsula as we hypothesize. This is just withing without a sea level change. You can see that it's more or less consistent. But we don't see it necessarily persisting over long periods of time like we had hoped, although you'll see a more nuanced view on that later. This is an animation of what that transect looked like on average over time in these simulations, kind of showing an initial strong influx of gene flow to the peninsula, but ultimately, as the source island got slowly swamped out, it declined. However, the focus of this is, of course, on that isolated nature of the peninsula. What we found was that when we have the black line, which represents the real map of Vanuatu, and the yellow line representing the one where I used Adobe Illustrator to fill in the Natewa Bay, we see even immediately there is a much different pattern where we don't see that sharp turn at that bend in the peninsula, or lack of peninsula in the other simulation. And this pattern lasts for a much shorter amount of time as well when we don't have a channel. You can see it play out here, right? How you kind of have an uptick that slowly turns into isolation by distance across Vanuatu over periods of time with this. But just these animations and whatnot aren't really sufficient for fully understanding this. So an undergraduate that I work with, Luis, made an algorithm where we can look at per replicate things of these kind of allele frequency curves with geography and identify an inflection point, not just once, but see how consistently it is. You're going to see that correlate with the size and transparency of dots. If we rarely find an inflection point at that point in the archipelago, it's going to be a smaller dot and more transparent, like you can see in this one here, which we might not infer any inflection point, but I wanted to put something there. Sorry. On the x-axis here, you're going to see the transect point like you did before. And the y-axis is just going to be going from the start of the simulation towards the end. You can see, as we saw in those summary graphs, we find many inflection points up until when you're on that peninsula, but once you get to the west of that bend, they kind of go away in our simulations with a channel. But when we have our channel filled in, we don't really see anything, right? We just see a little uptick right at the very end of that transect, as we predicted earlier. And going through time, it kind of holds steady for some amount, but over longer periods, it just kind of becomes more diffuse. Even though if you squint, you can kind of see more of a uptick towards the end of that channel, or sorry, when you have that channel, it really goes away. And as I mentioned, not the change over time, but the relative pattern that you see with and without the channel is what we expected, which is exciting. I also want to mention a way that I'm extending these simulations. So there are pretty much two hybrid zones in South Pacific Islands in birds. There's the whistlers and the Maizamala honeyeaters on the island of Makira, where they meet and actually can coexist in some capacity, forming some hybrids, but with strong reproductive isolation and assortative mating. And conveniently, they are very well worked on from ecological standpoint by Al Uy and Elsie Shogrim. So I'm working with them to kind of build them into a SLIM simulation and then swap the geographic context and see what happens. But I didn't want to go through those preliminary results today because they're pretty complicated. Now I want to talk a bit about the genomics of this divergence and this geographic radiation. This is a result from RADseq where we found one big peak correlated with population divergence, which encapsulates a gene in the melanocortin pathway that has been shown to correlate, whose transcription has been shown to correlate with barn owl plumage differences. However, this isn't entirely satisfying on its own, so we wanted to do a genome-wide association study. Currently, it's just with what we have, which is pretty low sample size, and for birds that are relatively unadmixed, meaning we don't have a ton of power to detect differences. But I wanted to try anyways, even before getting my new samples from fieldwork. And pretty much all that another undergraduate, Owen Peck, did to do this was in ImageJ, draw a transect across the extent of the band on these photos of specimens like this, and then look at the proportion of that little transect that is occupied by dark feathers. And we saw that it worked great, right? We identified a pretty good range of variation, although it's not totally bimodal. There's a bit of overlap. Very satisfactory for a first pass, despite our relatively low power. You see slightly different results when you look across scale. When you consider all yellow-throated taxa, including the ones that don't have immediate exposure to those intermediates, we see a peak on the Z chromosome. I'm also going to show you what the actual phenotypes are for different allele frequencies. So this is the Z peak. We find that you can see some amount of correlation within the focal populations on Vanuatu. The ones that are mostly unbanded are in yellow. You can see that it's slightly higher for the heterozygotes in the middle. And you can see that for the populations that are more banded, it's slightly lower for the heterozygotes. But ultimately, it's a relatively weak correlation, and you don't see it popping out as a significant GWAS hit in the bottom figure. Rather, the highest peak there is in a different region in an autosome that doesn't come out as a super strong FST peak, although you can still see signal of it in the other GWAS. And if you squint at FST plots that aren't here, you can make out something. And you do see a more strong relationship between it, although it is certainly not all of the story. There is one bird that has two homozygous dark banded alleles that you can see is kind of 50-50 or less on the amount of band extent on it, which tells us that this is likely pretty polygenic, and we really need this sampling from the core of the hybrid zone to identify these genes. But my interest in these genes isn't just finding things that correlate with melanin-related trait. I want to see what they're doing across the rest of the radiation. So when we look at these peaks and compare it to a genome-wide phylogeny, do we see elevated rates of taxon-specific evolution, suggesting kind of convergence in this area, driving the plumage divergence that we see across the radiation? Or do we see that it's an entirely different topology, either due to sorting of shared ancestral variation or potentially introgression? this is something that I haven't fully started yet, but I wanted to tease it while I'm here. And also note that we're using more specimens to calculate the actual band extent rather than doing that side-to-side transect. We're doing it across the whole length of the bird to get what proportion is occupied by a band. And you can see that we have a pretty good amount of variation with Fiji having relatively narrow bands compared to the rest, and a good amount of variation, hopefully enough to give us some power for signal. I'll also note this is not the full topology that we're going to wind up with. This is just what we already had sequences for. And this also is interesting for other aspects of plumage variation, right? There's a lot of variation in sexual dimorphism in this group. You see ones with very strong dimorphism with bright males. You see ones with very modest dimorphism with bright males. You see ones where both males and females are dull. Not pictured here. There's some where both males and females are incredibly bright, actually for arguably the brightest bird in the genus. Males and females are both spectacular looking and almost identical. Anyways, with time running low, I wanted to wrap this up with our conclusions. First, about what the dynamics of population genetic structure and genetic diversity are in the Solomon Islands. We found that dispersal ability is a key predictor of genetic divergence and likely gene flow when you look across the whole archipelago. And when you look at the relationship between island size and genetic diversity, it's sometimes pretty strong, but seems to be really dampened by ongoing gene flow within these island groups. When we look at Fiji to understand the generation of phenotypic diversity in these radiations, we find that plumage seems to evolve very rapidly during the colonization process, at least in this relatively dynamic group, but that it doesn't necessarily prevent gene flow, such as in this system. That geography is key in shaping both that hybrid population that we see and the hybrid zone on the peninsula. And with that, I would like to thank all the museums who made this possible, in addition to everyone who helped with field work, both for my own stuff in Fiji and all the work in the Solomons through Burke Museum, University of New Mexico, and University of Kansas, and the American Museum. And I'd also like to thank all of my collaborators from Texas Tech, Cornell, Kansas, University of South Pacific, UNM, and of course my funding sources, particularly the NSF PRFB that I have now. With that, I'll take any questions.
Your presentation, have you decided what are you going to use to analyze your last chapter with the phenotypic variation across the phylogeny?
Good question. It's something that I haven't fully started exploring yet. From what I've been seeing from other people, ABSREL seems to be pretty promising, although I'm also going to look at more simple metrics like DNDS, particularly before, like, not to do a genome scan type thing, but for those focal regions, just because I have this kind of a priori set, I can get around some of the power issues that we might have with other metrics. Yeah.
So I didn't quite get the MC1R stuff because these birds have black other than the band, so, right? So it's not like black versus no black knocking out MC1R. So what's going on with MC1R?
So for these ones in particular, MC1R isn't really an outlier. The paper that I mentioned wasn't. There are other genes related to the expression. I realize that's a bit misleading, but pretty much nothing's going on with MC1R in particular. It's, what is it? PSMK5, I think is the name. It's a much less frequently implicated gene in melanogenesis. And it's certainly not complete on its own, right? And in that chromosome 3 GWAS outlier, we don't actually see anything that has obvious plumage correlations in other taxa. I want to wait a bit more to really draw many conclusions about what exactly things are doing, but these are certainly like they appear to be pretty polygenic things. How many genes? Possibly pretty limited, but yeah, it's a more complex phenotype than that. And the exact way that that's evolving, it's a bit unclear if there's kind of universal sexual selection, universally beneficial sexual selection, or just kind of runaway processes going on. Tough to say for now, but it's an area that I'm pretty interested in. Tough to ask questions about, though, especially for these more complicated melanin traits. Oh.
For your island size diversity where the correlation is same as the flow, what is the exact nature of that decoupling? Is it that the small islands have more diversity than expected based on their size?
Yep, that's the main driver. You don't really see an elevated, like, you don't see a reduced diversity on the large islands. It's more like everything becomes homogenized. Looking at the raw values, yeah, it's pretty similar to the starting population in those larger islands, and it's just the smaller ones. And a lot of it is driven by the fact that those small ones, when they don't have ongoing gene flow, so quickly drift towards lower genetic diversity if you don't have ongoing gene flow. Yeah. Okay. Did you have a question first? Yeah.
Oh, okay, so pretty neat the way that you did, just like showing the changes to slope. I was wondering if you could do the same, like you predicting certain topologies across the year percent.
Great question. So that is something that I have done for another part of my work, kind of seeing the impact of gene flow on phylogenetic inference based on archipelago geography. And you certainly can. It's something that's relatively tricky to map onto a figure, but yeah, if you're interested in seeing some of that, none of it's published yet, but I'd be happy to show you some stuff. Yeah.
So when you're looking at this band extent on specimens, comparing it to length of bird, how are you controlling for like preparation type? Like some people make their necks really long.
That is absolutely a big issue with it, and one that's frankly tough to control for. Ultimately, it's a trait that we are kind of settled on being relatively noisy. And what I'm trying to do is get photos of birds from different predators in hopes that it kind of evens out at least across the tree. But yeah, it's something that's really hard to like fully address. Like we have a pretty standard protocol of like aiming to end it around a certain point in the specimen and start it at the chin, but there's certainly a lot of variation. I think the specimen photo that I actually showed, the pectoralis, is arguably showing more band than it should, for example, a relatively long neck. Yeah. And we certainly see this in especially some of the more extensively banded ones, where just a slightly longer neck just has much more of an impact, where it's relatively stable for like those Fiji ones. Big issue with it though. I mean, for the modern Fiji ones, conveniently, because they were mostly collected by Mike Anderson during his PhD, it's mostly prepped by the same person. And I do think that kind of going across the band works a little bit more consistently, but is still, hard. It's hard to find a perfect way of measuring pattern, particularly ones like this. But I'm pretty satisfied with it as a starting point, at least. Yeah.
Thought a lot about dispersal on Pacific Islands, and it seems to me no sane bird would leave an island. And so it strikes me as things like cyclones would be more likely to sort of cause dispersal than willingness to fly across open water. And then associated with that would be potentially birds that are roosting higher up in the canopy might be more prone to being blown off the island than birds that are lower down the canopy. Do you think that's any merit to that?
I haven't thought about the canopy height. The influence of cyclones is definitely one that I consider to be like a big outlying factor in this. Like my general thought is that it should even out over time, although in the paper where I described the kind of linking these island parameters to gene flow, the one outlier edge that we found was one that tracked trade winds in Fiji. Hard to know if it's perfect given the challenges of demographic modeling and gene flow, but it is at least one data point potentially pointing to the role of winds in all of this that is not currently being accounted for in the simulation. As for like canopy, though, it's definitely complicated. There's certainly many birds that are understory, but still do have kind of relatively little population structure across the archipelago. And I mean, even getting dragged across the whole Pacific in the cyclone seems unlikely, but we know that birds get from, say, Solomon Islands to Vanuatu to Fiji to Samoa, etc. So there is clearly some what's the term, Zugenroot going on there still, right? And I don't actually know. That makes me wonder. One system that has the best evidence for these long distance dispersal events is actually Darwin's finches, right? That famous big bird hybrid lineage is the result of a very long distance colonization. I don't have a sense for if they think that that was mediated by a cyclone or what, but yeah, it's definitely a hard thing to fully account for. And both trade winds and cyclones are two major unaccounted for factors here. And I'll mention doing it outside of birds on taxa that are more likely to raft. That also makes it a lot harder because you have to model potentially groups of organisms going across in this very rarely documented process. So yeah, figured I'd shout out rafting a bit.
Any last questions? If not, then feel free to join us on the front steps for our weekly museum seminar social and give one last round of applause to Ethan.
Thank you so much.